Hi, today I'm going to be talking about our experiment where we wanted to study whether acid rain would affect the growth of various plants. We looked specifically at Allium fistulosum, or the green onion. In order to conduct this experiment, we planned out that we would have 3 acidic solutions, so neutral pH of 7. First, we created stock solutions, which we could then process to get the specific pHs we needed. To make the stock solutions, we prepared 1 molar nitric acid and 2 molar sulfuric acid solutions. We started by adding about 800 milliliters of distilled water to the container. We slowly added 111.1 milliliters of concentrated sulfuric acid while stirring, then carefully added 62.5 milliliters of concentrated nitric acid. We stirred thoroughly to mix and topped off with distilled water to the 1,000 milliliter mark. We found the specific pHs using a pH probe, 5.5, 4.5, and 3.5 in pH, and then our control would be of solutions when we watered. In order to ensure there was data to collect, we waited to start watering with the pH solutions until we had some germination. We watered each plant with 30 milliliters of the assigned solutions. Once we started using the pH solutions, we began measuring the plants. For each pH group, there were 36 cups. Each cup was given a number 1 through 36. For each cup, the longest leaf was measured in millimeters and the data recorded. On an online spreadsheet, each day of measuring was placed on a separate tab with a column for each treatment and rows labeled 1 through 36. We did not receive growth in every cup, so those spots were kept empty. After reaching the end of our watering time, we pulled each plant from the cups as carefully as possible so as to not break the roots. We then ran our fingers along the length of the plant in order to get rid of as much dirt as possible. The plants were then placed on 4 different paper towels, 1 for each experimental group. We then used the dehydrator in order to dry out the green onions and collect a final dry biomass. The plan was to leave the green onions in the dehydrator for 24 hours, but then we lost power 4 to 5 hours before the green onions were to be collected. The power came back on later that day, but the green onions were not collected until the next morning. They ended up drying out for over 30 hours rather than the planned 24. Once dried, the green onions were placed on an electric balance inside of the paper towel. After being measured, green onions were taken out and the paper towel weighed. The mass of the paper towel was then subtracted from the initial mass taken to find the final dry mass of each experimental group. Since each group did not end with the same number of plants, the final dry mass was divided by the number of samples per category for an average dry mass. From the data collected, 3 graphs were created. The first graph we created was looking at the percent germination after we began our treatments. After we finished our treatments, we subtracted the total number of germinated plants we began applying our treatments from the total number of plants per treatment. We then divided each number by 36, which was the potential number of plants that could have grown. We found that our 7 pH had the highest percentage of germination after treatment began with about 81%, and 3.5 had the lowest with about 39%. We then looked at the average height of each group per recording day. The standard error bars on the graph were calculated from the recorded values per day per treatment. The 7 pH group had the largest average height on our last day of recording, even though it initially started with 0% germination. However, our 5.5 group and 3.5 groups had a similar pattern of height for the last 3 days of recording measurements, with their standard error bars overlapping. 4.5's growth stayed fairly constant throughout the experiment, with its average height only being second to the 7 group. These results are supported by an experiment conducted by Ramos et al. In their experiment, they looked at Monstera deliciosa and how various pH levels of water would affect their average absorbance of light. They found that as the pH of water decreased, the absorbance levels decreased. With our green onions, if the absorbance levels decrease, it is harder to take in light and therefore harder to conduct photosynthesis in order to grow. Therefore, as the pH decreases, the average height should as well. The average biomass was then recorded in an Excel spreadsheet and a graph created. The final average biomass of the 7 group was the largest value and 5.5 was the smallest, with 3.5 being close to it. These results are also mostly supported by a meta-analysis conducted by Shi et al. In an experiment conducted by Lal. In the meta-analysis, they looked at 69 different articles and found that across 51 different species, the average biomass of plants decreased as the pH of their simulated acid rain decreased. Lal's experiment saw similar results, but specifically in the mass of sunflower roots, shoots, and leaves. However, one part of our experiment that goes against the studies is our 4.5 group having a larger average dry mass and average height than our 5.5 group. While we cannot be certain what the cause was and would have to conduct more testing, we believe one explanation is that the specific ratio of acids used at that specific pH could have potentially created more available nutrients for those plants. So why does this matter? As we continue to burn fossil fuels and use pesticides or herbicides in agricultural practices, we are contributing to various forms of pollution. These kinds of pollution increase the amount of sulfur dioxide, nitrogen oxides, and organic acids found in the air. As these chemicals interact with ozone in the atmosphere, it makes the rain more acidic. Experiments like this one demonstrate the potential side effects of not changing our behavior. In our experiment, we used Allium fistulosum, or the green onion, as a stand-in for various other native species like Allium munzi and Allium trichocum. This experiment shows the potential harm that native species may face if we cannot reduce our part in creating acid rain.